Number 1. Methanosarcina. In 2014, Daniel Rothman and Gregory Fournier at the Massachusetts Institute of Technology proposed something far stranger. The real killer, they argued, was not a volcano. It was a single-celled organism smaller than a red blood cell. Methanosarcina, an archaeon. Not even a bacterium, but a member of a separate domain of life entirely. And it had a weapon it had recently stolen. Molecular clock analysis showed that sometime around 252 million years ago, Methanosarcina acquired a gene from Clostridia bacteria through horizontal gene transfer, a scrap of genetic code jumping between two completely unrelated lineages. That gene unlocked a metabolic pathway called acetoclastic methanogenesis, the ability to convert acetate, a form of organic carbon that had been accumulating in ocean sediments for millions of years, directly into methane and carbon dioxide. Before this gene transfer, those acetate reserves had no efficient biological pathway to reach the atmosphere. Afterward, Methanosarcina could eat them, but the gene alone was not enough. The Siberian Traps eruptions, which lasted approximately 1 million years, released massive quantities of nickel into the environment. Nickel is an essential cofactor for the enzymes that drive methane production in archaea. Nickel concentrations in sedimentary rocks spike precisely at the Permian-Triassic boundary. The volcano provided the catalyst, the gene provided the capability, and Methanosarcina did what any organism does when handed unlimited food and a growth accelerant. It bloomed. Rothman's mathematical model showed that the resulting carbon release was super exponential, faster than any purely geological process could account for, but perfectly consistent with the growth dynamics of a microbial population doubling and doubling again. The methane oxidized to carbon dioxide in the atmosphere within decades, but the carbon dioxide persisted for millennia, driving sustained ocean acidification that dissolved the calcium carbonate shells of marine invertebrates. Global mean temperatures rose by an estimated 8 to 10 degrees. The competing view, championed by researchers like Seth Burgess, holds that Siberian Traps volcanism alone is sufficient, but Rothman's counter-argument is pointed. The carbon isotope excursion at the boundary is too large and too rapid for volcanism alone to explain. Something biological was amplifying the signal, and Burgess's own uranium lead dating showed the extinction lasted approximately 60,000 years. That is too fast for geology. It is exactly right for biology. Methanosarcina species are still abundant today. They thrive in rice paddies, in the guts of ruminants, in sewage treatment plants. They produce roughly 60% of all biogenic methane on Earth, and the gene they stole from Clostridia 252 million years ago is still sitting in their genome, a permanent record of the most destructive biological event in the history of complex life, a heist written in DNA. But Methanosarcina did not work alone. The end Permian was a cascade, and the next organism in that cascade made sure that even after the killing stopped, nothing could come back. Number 2. Toxic microbial blooms. After 96% of marine life and the vast majority of terrestrial forests were gone, you would expect recovery to begin. Ecological niches were empty, resources were available. The survivors should have radiated, filled the gaps, rebuilt. Instead, for over 100,000 years, life could not come back. Something was actively holding the planet in a state of death. In 2021, Chris Mace, Stephen McLaughlin, and their colleagues published their findings in Nature Communications. Working with sediment cores from the Sydney Basin in Australia, one of the best preserved Permian-Triassic freshwater records on Earth, they identified the culprit. Toxic microbial blooms, cyanobacteria, and microbial mat communities that had colonized freshwater systems across the planet and were producing hepatotoxins and neurotoxins at concentrations sufficient to kill anything that tried to live in or drink from those waters. The trigger was deceptively simple. The end Permian extinction destroyed forests. Without root systems holding soil in place, an estimated 95% of terrestrial soil cover eroded into rivers and lakes. That soil carried phosphorus and nitrogen, the essential nutrients for microbial growth, in quantities tens to hundreds of times above normal. Simultaneously, atmospheric carbon dioxide levels were elevated and global temperatures were 8 to 10 degrees above normal. Warm, nutrient-rich, stagnant water. These are the exact conditions that modern limnologists identify as ideal for harmful algal blooms. Except at the end Permian, these conditions existed everywhere, globally, for tens of thousands of years. The blooms created a feedback loop that was nearly impossible to break. They killed aquatic organisms. The dead organisms decomposed, releasing more nutrients into the water. Those nutrients fueled more blooms. The cycle repeated, and because the blooms preferentially colonized shallow water environments, they occupied exactly the ecological space where recovery would normally begin. They placed a dead zone at the starting line. Sebastian Grasby and colleagues showed in 2020 that coal fly ash from the Siberian traps, which had burned through carboniferous coal seams during the eruption, provided additional nutrient loading. The volcano was fertilizing the blooms from above. Recovery was episodic. Sediment cores show alternating layers, brief windows where the blooms receded and organisms began to reappear, followed by bloom recurrence and another wave of death. Freshwater vertebrate diversity in the Sydney Basin did not recover to pre-extinction levels for approximately 3 million years, and similar bloom signatures have been found in contemporaneous deposits in South Africa, China, and Greenland. This was not a local anomaly, it was planetary. The term used in the literature is malignant bloom. These organisms were not passively occupying space. They were actively suppressing recovery through toxin production, light blocking, and oxygen depletion. And the modern parallel is not theoretical. Harmful algal blooms are increasing worldwide. Lake Erie, the Baltic Sea, Lake Taihu, the Murray-Darling Basin. The same mechanism, at a smaller scale, is already running. But the toxic blooms were not the only microbes weaponizing the end Permian Ocean. Another group of organisms turned the water itself into poison gas. Number 3, Treptichnus Pedum-grade burrowing worms. Before the end Permian, before the first dinosaurs, before anything with a backbone had walked on land, there was an even stranger world. The Ediacaran period, 575 to 539 million years ago. The organisms that lived then look like nothing alive today. Rangeomorphs, fractal fronds anchored to the seafloor like alien ferns. Dickinsonia, flat-quilted ovals that may have absorbed nutrients directly through their skin. Kimberella, a slug-like grazer. They were soft-bodied, mostly immobile, and they lived on thick microbial mats that carpeted the seafloor in laminated layers of photosynthetic bacteria. Those mats were everything: foundation, food source, oxygen supply. Then something evolved that could dig. Treptichnus pedum is a trace fossil, not the organism itself, but the burrow it left behind. The actual creature was likely a priapulid or priapulid-grade worm, soft-bodied, probably no more than a few centimeters long. But the trace fossil it left is so significant that it formally defines the base of the Cambrian period. The first appearance of Treptichnus pedum is by International Geological Convention, where the Cambrian begins. What these worms did was not dramatic in the way a volcanic eruption or an asteroid impact is dramatic. They burrowed. They pushed through the microbial mats, broke them apart from below, oxygenated sediment layers that had been anoxic for billions of years, mixed the upper layers of the seafloor into an unstable, churned substrate. Paleontologists call this the agronomic revolution. Others call it the Cambrian substrate revolution. Simon Darroch and colleagues demonstrated in 2015, working with fossils from the Nama group in Namibia, a direct statistical correlation between increasing bioturbation intensity and declining Ediacaran biodiversity. As the burrowing got more complex, the Ediacaran organisms disappeared. The Ediacaran biota had no defenses, no shells, no ability to burrow themselves, no mobility in most cases. They were anchored to a substrate that was being systematically destroyed, and the worms did not need to eat them to kill them. They just needed to destroy the mat. Marcos Mangano and Luis Boitoy documented the escalation. Simple horizontal traces gave way to three-dimensional burrow networks. Each increase in burrowing complexity corresponded to another drop in Ediacaran diversity. The extinction took roughly 10 million years, not a sudden catastrophe. A slow ecological squeeze. An estimated 80% of Ediacaran genera disappeared. Some survivors, like Swartpuntia, persisted briefly into the earliest Cambrian before vanishing entirely. The mainstream view, supported by Douglas Irwin, Darroch, and others, holds that this was the first mass extinction driven primarily by biological innovation rather than environmental catastrophe. A modern analogy exists. Invasive earthworms in North American boreal forests are currently destroying the leaf litter layers that entire woodland ecosystems depend on. The mechanism is identical. Burrowing organisms dismantle the substrate, and everything that depends on that substrate collapses. The first complex ecosystem on Earth was not destroyed by fire or ice or poison. It was destroyed by worms that simply dug through the floor. Number 4. Sulfate-reducing bacteria. Returned to the end-Permian ocean 252 million years ago. Methanosarcina has already triggered catastrophic warming. The global temperature has risen by 8 to 10 degrees, and in the deep ocean, something else is happening. The warming has disrupted ocean circulation. Warm surface water sits on top of cold deep water like oil on vinegar. The layers do not mix. Oxygen from the atmosphere cannot reach the deep ocean. And as the deep water becomes anoxic, oxygen-free, a different kind of life takes over. Sulfate-reducing bacteria, Desulfovibrio, Desulfobacter, Desulfococcus. These are anaerobic microorganisms that breathe sulfate instead of oxygen, and their waste product is not carbon dioxide, it is hydrogen sulfide, the gas that smells like rotten eggs at low concentrations and kills by paralyzing the nervous system at high ones. In the modern ocean, sulfate-reducing bacteria are confined to small anoxic basins, the Black Sea, the Cariaco Basin off Venezuela, pockets. During the end Permian, the entire deep ocean may have gone anoxic, a euxinic ocean, and the sulfate-reducing bacteria filled it. Lee Kump, Alexander Pavlov, and Michael Arthur published the hydrogen sulfide ocean hypothesis in Geology in 2005. Their model shows the mechanism. As the deep ocean became euxinic, hydrogen sulfide concentrations rose until the gas reached the photic zone, the sunlit upper ocean, and began escaping into the atmosphere. The biomarker evidence is unambiguous. Isoranierotuni, a pigment produced exclusively by green sulfur bacteria that require both sunlight and hydrogen sulfide, has been found in Permian-Triassic boundary sediments on every continent. These bacteria could not have existed unless hydrogen sulfide was present in the sunlit surface waters. The gas was everywhere. Atmospheric hydrogen sulfide at concentrations above roughly 200 parts per million kills most terrestrial animals within minutes. But the killing did not stop there. Hydrogen sulfide reacts with ozone. It breaks down the ozone layer, producing sulfur aerosols that thin the shield against ultraviolet radiation. With the ozone compromised, UVB radiation flooded the surface. Evidence exists in the rock record. Mutated spores called teratological polynomorphs appear at the Permian-Triassic boundary worldwide. The deformities are consistent with the kind of DNA damage caused by intense ultraviolet exposure. Kump's model estimated atmospheric hydrogen sulfide concentrations of 200 to 2,000 parts per million in coastal regions. This explains a puzzle that had troubled paleontologists for decades. Ocean anoxia kills marine life. But the end Permian killed enormous numbers of land animals too. Why would oxygen-depleted oceans affect creatures living on land? The answer is that the oceans were not merely losing oxygen. They were producing a gas that was toxic, heavier than air, and capable of flowing across coastlines like an invisible flood. Peter Ward of the University of Washington has spent years championing this hypothesis, arguing that hydrogen sulfide events have occurred during multiple mass extinctions, not just the end Permian. The sulfate-reducing bacteria did not cause the initial warming. They did not cause the anoxia. Those were triggered by volcanism and amplified by Methanosarcina. But the bacteria transformed a warming event into chemical warfare. They took an ocean that was losing oxygen and turned it into an ocean that was actively producing poison. And the terrifying detail is this: these bacteria are still everywhere. In every anoxic sediment, every waterlogged soil, every sewage system, every dead zone in every ocean on Earth. The only thing preventing a repeat is ocean circulation, keeping the deep water oxygenated. Number 5. Reduviasporonites. After Methanosarcina triggered the warming, after sulfate-reducing bacteria turned the ocean to hydrogen sulfide, after toxic microbial blooms colonized the freshwater and blocked recovery, after 96% of marine species and 70% of land vertebrates were dead, something else moved in. Something grew on the corpse of the world. In rock sections from the Permian-Triassic boundary, across every continent on Earth, the microfossil record tells the same story. A single organism dominates. Greater than 95% of all polynomorphs, the microscopic organic walled fossils that paleontologists use to date and correlate rocks, belong to one thing: Reduviasporonites. Branching filaments with thick, dark walls. First described by Elsick in 1999. Identified as fungal by Henk Vischer and his colleagues at Utrecht University in the late 1990s. Confirmed on every continent. A global biological takeover. The identity was contested. Mark Sefton at Imperial College London argued in 2009 that the carbon isotope ratios were inconsistent with fungal tissue and that Reduviasporonites might be algal. Vischer and colleagues responded in 2011. A saprotrophic fungus feeding on isotopically light dead wood would produce exactly the carbon isotope signature Sefton observed. The isotopes reflected diet, not taxonomy, and chitin, the polysaccharide that forms fungal cell walls, was subsequently detected in Reduviasporonite specimens using Fourier transform infrared spectroscopy. The consensus now leans fungal, though some residual debate persists. The ecological picture is stark. The end Permian killed an estimated 95% of terrestrial plant life. Forests that had covered much of Gondwana and Laurasia were dead. The resulting biomass, billions of tons of dead wood and leaf litter, accumulated on a planetary scale with no herbivores to consume it and no competing decomposers to break it down. Reduviasporonites filled the vacuum. It colonized this dead biomass, and in the low oxygen early Triassic atmosphere, estimated at 15 to 16% oxygen compared to the modern 21%, it thrived. Many saprotrophic fungi are facultatively anaerobic. Low oxygen was not a barrier. It was an advantage, eliminating competitors. But was Reduviasporonites merely a decomposer, or was it something worse? Henk Vischer and Cindy Louie found fungal hyphae penetrating what appeared to be living plant tissue in Permian-Triassic boundary fossils. This suggests the fungus was not only feeding on the dead, it may have been actively infecting and killing the weakened survivors, preventing the forests from regrowing. If true, Reduviasporonites was not just colonizing a dead world, it was holding the world dead. Cindy Louie at the University of California, Berkeley, has documented the aftermath. Plant community recovery after the fungal spike was extraordinarily slow. The coal gap, a period of approximately 10 million years after the Permian-Triassic boundary, during which no coal formed anywhere on Earth, is one of the most striking signatures in the geological record. Coal requires forests. Forests did not exist at scale for 10 million years. Reduviasporonites is now used as a stratigraphic marker. The fungal spike is so distinctive and so globally consistent that geologists use it to correlate rocks between continents, the same way they use the iridium anomaly to mark the asteroid that killed the dinosaurs. A modern analogy exists. Armillaria ustoyae, the honey fungus, is the largest known organism on Earth, a single individual spanning approximately 10 square kilometers in the Blue Mountains of Oregon, killing trees across its entire extent. But, at the end Permian, the entire planet was a substrate. Number 6. Early mosses. 445 million years ago. The late Ordovician, the second most severe mass extinction in the Phanerozoic eon. 85% of marine species, approximately 60% of marine genera, gone. Trilobites, brachiopods, bryozoans, graptolites, decimated. The killing mechanism was glaciation. Ice sheets expanded from a south pole, then located over what is now North Africa and the Sahara, spreading across much of the supercontinent Gondwana. Sea levels dropped, shallow marine habitats where the overwhelming majority of marine life existed were destroyed. The question is what caused the glaciation, and in 2012, Tim Lenton at the University of Exeter provided an answer that almost nobody expected. Moss. The first land plants were non-vascular bryophytes, liverworts, hornworts, and mosses. They began colonizing bare rock surfaces approximately 470 million years ago in the mid-Ordovician. They had no roots. They used rhizoids, fine hair-like filaments that penetrated rock surfaces and extracted minerals. They were small, inconspicuous, and they were destroying the atmosphere. The mechanism is called biotic weathering. When bryophytes grow on silicate rock, their rhizoids and the organic acids they produce accelerate the chemical breakdown of that rock. The weathering reaction consumes atmospheric carbon dioxide. Calcium silicate plus carbon dioxide yields calcium carbonate plus silica. More weathering means less carbon dioxide in the atmosphere. Lenton's model, published in the Proceedings of the National Academy of Sciences, estimated that bryophyte-enhanced weathering increased the rate of silicate breakdown by a factor of 3 to 8 compared to bare rock. Over 25 to 30 million years, this drew down atmospheric carbon dioxide from approximately 4,000 parts per million to approximately 2,200 parts per million, a reduction of roughly 50%. This was enough. The Earth's climate tipped. The Hernantian glaciation began. Ice sheets advanced. Sea levels fell by an estimated 50 to 100 meters, and 85% of marine species died. Jonathan Quirk and colleagues provided experimental confirmation in 2015, growing modern bryophytes of the species Fiscometrella patens on silicate rock under controlled laboratory conditions. The measured weathering enhancement was 3 to 8 times faster than abiotic weathering, matching Lenton's model precisely, and the plants did not need to cover vast areas. Lenton's calculations showed that even 5 to 10% coverage of available rock surface was sufficient because the per-area weathering enhancement was so large. The extinction itself had 2 pulses. The first, driven by glacial onset, destroyed shallow marine habitats through cooling and sea level drop. The second, triggered by glacial termination, brought rapid warming, ocean stratification, and anoxia. Mosses may have contributed to both. The carbon dioxide drawdown drove the first pulse. The weathering products, nutrients washed into the ocean, may have fed the anoxia of the second. Phil Jardine at the University of Münster has used biogeochemical models to independently confirm that mid-Ordovician plant colonization could have reduced atmospheric carbon dioxide by 30 to 50% within 20 million years. The tectonic counter-argument that Gondwana's position over the South Pole was sufficient to explain the glaciation does not hold on its own. The continent had been in roughly the same position for tens of millions of years before the glaciation started. Something changed. That something, Lenton argues, was the colonization of land by moss. The irony is absolute. The organisms that would eventually make land habitable for all future terrestrial life by creating soil and drawing down carbon dioxide to temperate levels, first had to trigger a mass extinction to do it. Number 7. Lepidodendron. 40 meters tall, 2 meters in trunk diameter. A bark surface covered in diamond-shaped scars, each marking where a grass-like leaf was once attached. Lepidodendron was a lycopsid, related to modern club mosses, which today grow no higher than your ankle. In the Carboniferous, 323 to 299 million years ago, they were giants. The growth pattern was alien compared to modern trees. Lepidodendron grew rapidly to full height, possibly in as little as 10 to 15 years. It branched only at the very top, in a narrow crown of dichotomous forks bearing strap-like leaves. It reproduced by spores, not seeds. And when it died, nothing could eat it. The bark and trunk of Lepidodendron were extraordinarily rich in lignin, a complex organic polymer that provides structural rigidity. In the modern world, white rot fungi are the primary decomposers of lignin. Their enzymes, lacases, and peroxidases break the polymer into digestible components. But in the Carboniferous, according to the hypothesis advanced by J. M. Robinson in 1990 and refined by Robert Berner in 2003, these enzymes had not yet fully evolved. There was a decomposition lag. Dead Lepidodendron trunks fell into the swamps, were buried by sediment, and instead of decomposing, they compressed. Over millions of years, under heat and pressure, they became coal. This is the origin of most of the coal on Earth. The Carboniferous period literally means coal-bearing. Approximately 90% of the world's coal deposits formed during this window, and every ton of that coal represents roughly 3.5 tons of carbon dioxide that was removed from the atmosphere and locked underground. The scale of carbon burial was staggering. Atmospheric carbon dioxide dropped from approximately 1,500 parts per million in the early Carboniferous to approximately 300 parts per million by the late Carboniferous, approaching modern pre-industrial levels of 280. Simultaneously, because all that carbon was being buried rather than oxidized, atmospheric oxygen rose to approximately 35% compared to the modern 21%. This is why Carboniferous insects grew to extraordinary sizes. Meganeura, a dragonfly with a 70-centimeter wingspan, could only exist in a high oxygen atmosphere. But the carbon dioxide crash had consequences. It triggered the late Paleozoic Ice Age. Ice sheets expanded across southern Gondwana, and the climate oscillated between glacial and interglacial periods with cycles lasting approximately 100,000 years. The Carboniferous rainforest collapse approximately 305 million years ago was the result. The glacial-interglacial oscillations fragmented the vast continuous tropical swamp forests into isolated patches. Amphibian lineages disappeared. Early reptile diversity plummeted. The giant arthropods, dependent on high oxygen and continuous forest habitat, eventually declined. Matthew Nelson and colleagues at the Field Museum challenged the lignin lag hypothesis in 2016, arguing that white rot fungi evolved in the late Carboniferous and that burial in anoxic waterlogged swamps was a more important factor than lignin resistance. But regardless of the mechanism, the result is identical. Unprecedented carbon burial, atmospheric carbon dioxide crash, glaciation, ecosystem collapse. The supreme irony of Lepidodendron is that its body could not be broken down, and that indestructibility made it the most effective carbon sink in Earth's history. It drew down carbon dioxide so efficiently that it froze the planet and destroyed the very forests it belonged to. The coal we burn today is literally the compressed remains of Lepidodendron and its relatives. We are releasing the carbon they sequestered 300 million years ago, reversing their work, and accelerating our own version of the story they already told. Number 8. Archaeopteris. This is where the story of trees destroying the world begins. Not in the Carboniferous swamps, but 50 million years earlier, in the late Devonian. 375 million years ago, when the first true tree evolved. Archaeopteris had a woody trunk with growth rings. It reached heights of up to 30 meters. It bore fern-like fronds and reproduced by spores, a progymnosperm, an evolutionary bridge between ferns and seed plants. And it developed something no plant before it had possessed: deep roots. Root systems that penetrated meters into the bedrock beneath the soil. Thomas Algeo at the University of Cincinnati and Stephen Scheckler at Virginia Tech published the key hypothesis in 1998 in the Philosophical Transactions of the Royal Society. The first forest, they argued, caused the late Devonian mass extinction, and the weapon was not the trees themselves. It was what their roots did to the rock. Root-driven weathering cracked bedrock and accelerated the chemical breakdown of minerals. Phosphorus, a nutrient that limits biological productivity in most marine environments, was released into river systems at rates estimated at 10 to 100 times pre-forest levels. The rivers carried this phosphorus to the coast, and in the shallow seas where sunlight and nutrients combined, algal blooms erupted at scales never seen before. When the blooms died, their decomposition consumed dissolved oxygen. The shallow seas, home to the overwhelming majority of marine life in the Devonian, became anoxic, dead zones. The geological evidence is written in black shale, organic-rich, oxygen-depleted sediment deposited during episodes of widespread anoxia. Black shale layers appear globally at the Kellwasser events, approximately 372 million years ago, and at the Hangenberg event, approximately 359 million years ago. These are the two main killing pulses of the late Devonian extinction. The Kellwasser events killed approximately 22% of marine families and up to 50% of marine genera. Reef ecosystems were devastated. The stromatoporoid coral reef system, which had dominated shallow marine environments for 100 million years, never recovered. George McGee at Rutgers University has documented that Metazoan reef complexity did not return to its former levels for approximately 100 million years. The reef gap. One of the longest ecological recoveries in Earth's history, Algeo's phosphorus hypothesis is supported by geochemical data showing spikes in phosphorus concentration in marine sediments that coincide precisely with the expansion of Archaeopteris forests across Laurasia and Gondwana. His model demonstrates that even 15 to 20% forest coverage of continental landmasses would have been sufficient to drive the observed nutrient pulse, and Archaeopteris was cosmopolitan. It grew everywhere. Competing hypotheses exist. The Vilui traps volcanism in Siberia, possible bolide impacts, intrinsic changes in ocean circulation. The consensus view is multifactorial, and Archaeopteris was likely one significant driver among several, not the sole cause. Jessica Whiteside and colleagues have used mercury concentrations in Devonian sediments to argue for a volcanic role, but the nutrient runoff signal from forests persists alongside the volcanic signal. Both were operating. The modern parallel is not subtle. The Gulf of Mexico hypoxic zone, approximately 15,000 to 22,000 square kilometers every summer, is driven by exactly the same mechanism. Nutrient runoff from agricultural land washes into the Mississippi River, feeds algal blooms in the Gulf, and creates a dead zone where nothing with gills can survive. Archaeopteris pioneered this process 375 million years ago. We perfected it. The end Permian was not one catastrophe. It was a biological chain reaction. Methanosarcina's methane triggered the warming. Warming caused ocean stratification and deep water anoxia. Sulfate-reducing bacteria colonized the anoxic deep ocean and produced hydrogen sulfide. The hydrogen sulfide escaped into the atmosphere, destroyed the ozone layer, and killed terrestrial life. With forests dead, soil eroded into freshwater systems. Toxic microbial blooms colonized the nutrient-rich water and prevented recovery for over 100,000 years. And Reduviasporonites colonized the dead biomass, possibly infecting and killing the survivors. Each organism amplified the damage done by the one before it. The cascade lasted approximately 60,000 years for the initial killing phase, plus 100,000 or more years of suppressed recovery. Full ecosystem recovery took approximately 5 to 10 million years, the longest in the Phanerozoic. The coal gap, 10 million years of no coal formation anywhere on Earth, testifies to the completeness of the destruction. Forests simply did not exist at scale for that entire period. The lesson is structural. Mass extinctions are not single events. They are cascades in which biological amplifiers take a trigger, usually volcanic, and magnify it into an apocalypse. And the modern relevance is not academic. Current warming is creating conditions for each of these organisms to thrive again. Ocean dead zones are expanding. Freshwater algal blooms are increasing. Methane emissions from microbes are accelerating. The same chain reaction, warming to anoxia, to microbial takeover to chemical warfare, is not theoretical. It has happened. The organisms are still here, still in every anoxic sediment and stagnant waterway, waiting for nothing more than the conditions they had before. 3 different plants, 3 different geological periods, one mechanism. Altering the atmosphere through rock weathering and carbon burial, mosses enhanced weathering by a factor of 3 to 8, crashed carbon dioxide by roughly 50%, and triggered the Ordovician glaciation. Archaeopteris developed deep roots that cracked bedrock, released phosphorus at 10 to 100 times normal levels, and triggered marine eutrophication and anoxia across the Devonian Ocean. Lepidodendron buried carbon as coal at a scale never seen before or since, crashing carbon dioxide from 1,500 to 300 parts per million and triggering the late Paleozoic Ice Age. Each plant was more powerful than the last. The evolutionary progression from rhizoids to shallow roots to deep roots to lignin-rich trunks represented an escalating capacity to modify rock, soil, water, and atmosphere. And every one of these plants eventually suffered from its own success. Mosses cooled the planet they were colonizing. Archaeopteris destroyed the marine ecosystems downstream of its forests. Lepidodendron froze the swamps it lived in. The pattern, ecosystem engineers destroying their own habitat through success, is one of the most persistent in Earth's history. And it reveals a counterintuitive truth. All 3 plant-driven extinctions primarily killed marine life, even though the plants themselves were terrestrial. Land organisms destroying ocean ecosystems through atmospheric and nutrient teleconnections. Modern agriculture continues this pattern. Fertilizer runoff from cropland creates marine dead zones by exactly the same mechanism Archaeopteris pioneered 375 million years ago. The Gulf of Mexico, the Baltic Sea, the East China Sea, these are the modern Kellwasser events. Every organism in this video is either microscopic or superficially harmless. Archaea, bacteria, fungi, moss, ferns, trees. Not a single apex predator, no tyrannosaur, no megalodon, no anomalocaridid, has ever come close to ending life on Earth. And this is not a coincidence. Predators are constrained by their prey. They cannot overshoot without destroying their own food supply. Negative feedback. Self-limiting. The organisms that almost ended life share a different trait entirely. They modify the environment itself. They are ecosystem engineers. And ecosystem engineering operates through positive feedback loops. Once started, the process amplifies itself. A predator kills one organism at a time. An ecosystem engineer changes the conditions for all organisms simultaneously. Methanosarcina changed ocean chemistry globally. Mosses changed the rock cycle across continents. Lepidodendron changed the carbon cycle for millions of years. Sulfate-reducing bacteria changed the composition of the atmosphere itself. The geological record is unambiguous. Biogeochemical disruption, not predation, drives mass extinction. The deadliest organisms in Earth's history are the ones you would never notice. They reproduce exponentially, operate at the chemical level, and when trillions of cells perform the same reaction simultaneously, the effect scales to planetary impact. They do not break the rules of the ecosystem. They change the rules entirely. Every organism in this video is still alive. Methanosarcina thrives in rice paddies, sewage digesters, and ruminant guts, producing roughly 60% of all biogenic methane on Earth. Sulfate-reducing bacteria inhabit every anoxic sediment, every waterlogged soil, every expanding dead zone in every ocean. Toxic cyanobacterial blooms are increasing across every continent, in Lake Erie, the Baltic Sea, Lake Taihu, the Murray-Darling Basin. The conditions that allowed each of these organisms to devastate the planet are returning. Ocean stratification is increasing. The deep ocean is losing oxygen at approximately 2% per decade, according to Sunka Schmidtko and colleagues in 2017. Marine dead zones have quadrupled since the 1950s. Agricultural nutrient runoff is driving freshwater and coastal eutrophication at rates without precedent in the geological record. Methane emissions from wetlands and permafrost are accelerating with every fraction of a degree of warming. Lee Kump has explicitly warned that modern ocean deoxygenation could trigger hydrogen sulfide production in deep basins within centuries. The Black Sea is already eosinic hydrogen sulfide rich below 150 meters, a small-scale modern analog of the end Permian Ocean. The same chain reaction, warming to stratification to anoxia to microbial takeover, is not a scenario. It is a mechanism that has operated before and whose components are all present today. The difference in speed is striking. The end Permian took roughly 60,000 years. Current carbon dioxide rises approximately 100 times faster than the end Permian rate. The organisms do not need to evolve, they do not need to adapt, they do not need to acquire new genes or find new niches. They just need the conditions they have been waiting for. Warmth, nutrients, anoxia. Every one of the catastrophes described in this video began with a slow trigger and ended with exponential biological amplification. The trigger this time is anthropogenic carbon dioxide. The amplifiers are the same organisms that almost ended life before. Over 4 billion years of life on Earth, the closest calls, the moments when the biosphere came nearest to total collapse, were not caused by asteroids or comets or gamma-ray bursts. They were caused by organisms doing what organisms do: metabolizing, growing, reproducing, modifying their surroundings. A worm that burrowed through a mat, a moss that weathered a rock, a tree that grew roots into bedrock, an archaeon that learned to eat acetate. None of them intended to destroy anything, they were simply successful. And their success, compounded over geological time and scaled to planetary surface area, was catastrophic. The line between ecological success and mass extinction is thinner than it appears. Every organism in this video crossed that line not through malice, not through competition, not through predation, but through metabolism. The chemical reactions that sustain life are the same chemical reactions that, at sufficient scale, can end it. Photosynthesis produced oxygen, a poison to 80% of the Archaean biosphere. Methanogenesis produced methane, a greenhouse gas that boiled the Permian Ocean. Decomposition recycled carbon until a fungus monopolized a dead planet. Weathering built soil and crashed the atmosphere in the process. These are not aberrations. They are not anomalies or bad luck or 1 in a billion coincidences. They are what life does. The geological record is not a catalog of unfortunate disasters. It is a record of biological power, power that life wields unconsciously and without restraint. The organisms that almost ended life on Earth are still here. They are in every soil sample, every water droplet, every cubic centimeter of ocean sediment. They are waiting for nothing. They are simply alive. And if the conditions return, the warmth, the nutrients, the anoxia, they will do exactly what they did before. Because that is what life does. It grows until something stops it.